name's McElroy. I'm going to be talking about comparing gene expression between sexual and asexual snails. Sexual reproduction is seen across the vast majority of plants, animals, and fungi. It is ancestral for the eukaryotes. And despite its commonness, it is associated with profound costs compared to asexual reproduction. And it is because of this that after decades of asking this question, we are still asking, why sex? Why has sexual reproduction become so common? And although we understand the nuts and bolts of sex fairly well, we understand meiosis, fertilization, and development, when it comes to questions about how sex arose and how it is into the maintenance of sex. Ideally, we can study organisms that exist as sexual and asexual individuals. And there are species that allow us to do this. In particular, Potamopergus antipodarum is a freshwater New Zealand snail that is characterized by multiple independent transitions to asexuality. So this is a phylogenetic tree of the species. Blue are sexual lineages, and red are asexual lineages. And this color scheme of the blue and red I'm going to keep throughout the presentation. And we know that with this species, sexual reproduction is ancestral, and that on occasion, at an unknown frequency, obligate asexual females will produce obligate asexual females. And we know that this there's a genetic component to this. It's a reliably inherited trait that has 100% penetrance and only goes one direction. The asexuals never revert back to sexual reproduction, and they coexist and compete in nature for the same resources, making this an excellent model to study sex and asex and the maintenance of sex. And it allows us to ask questions like what the genomic and genetic factors involved in frequent transition to asexuality are. So these types of questions include what the types of genes involved with the transition are. We know that with species like the monoclinic rotifers, there's a single locus involved with the transition to asexuality, whereas in Daphnia, it's a complex trait involving at least 4 loci, and we don't really know how this happens in this species. So what we don't know, like I said, causes. We don't really know what accounts for these transitions to asexuality. Furthermore, the genetic and genomic consequences of transition to asexuality are still unclear. We aren't really sure what genes are most affected by asexuality. We don't know quite the global, the genomic consequences either. Another area that we're interested in studying are whether these are repeated or unique factors involved with each transition, or if it's unique every time. And then if the evolutionary trajectories that these asexual lineages follow, if these are kind of predictable or if they're unique every time. So an approach that we've taken to start trying to answer these questions involves gene expression of sexual and asexual snails, comparing these. And we hope with this approach that we can start to identify loci that are expressed differently between the sexual and asexual snails, and start to develop sets of genes that are likely to be involved with these transitions and are most influenced by this mode of reproduction. And so what we've done is use ovarian tissue. This is a site of egg production. This is kind of what we thought would be a good start for trying to characterize the genetic, the differences in gene expression between sexual and asexual snails. So what we did was use an RNA-Seq experiment. So we extracted RNA from ovarian tissue of actively reproducing females. So these were brooding females. They were producing offspring at the time that we dissected them and then extracted DNA, or RNA, sorry. We used 2 biological replicates from 3 sexual and 3 asexual lineages of snails. And we used Illumina paradigm sequencing to generate millions of reads of RNA. And the Trinity assembly program to develop a de novo transcriptome for the for ovarian tissue, which we use as a reference in the tuxedo pipeline for differential expression analysis, which you take two conditions and you take RNA reads from those two conditions and map them against a reference genome, or in our case, the reference transcriptome, which we use we pooled our sexual and asexual reads into these two conditions, map them against the transcriptome, and then we're able to do differential expression analyses, and then use the R package cummerbund to visualize these expression profiles. So, for example, this is a heat map produced in cummerbund, and each row corresponds to a unique transcript, and the color over here represents the level of expression. So higher, the darker the color, the higher the expression. And of almost 39,000 transcripts that were tested for differential expression between sexual and asexual snails, we saw that over 360 were expressed higher in asexuals, and a little over 200 were expressed higher in sexuals. So we thought it would be worth taking a look at every pairwise comparison of these sexual and asexual lineages, because, like I said earlier, these asexual lineages are the result of independent transitions to asexuality. They aren't a clade, essentially. So what we have here are each possible comparison of a sexual to an asexual lineage, and we see that generally the asexuals have a higher amount, have more transcripts that are expressed at a higher level than the sexuals, and these are all these transcripts in these sets were expressed significantly higher. And this pattern is significant with the Wilcoxon sign rank test. And since we see a fair amount of variation in the amount of differentially expressed transcript, we thought we'd look at the heat map again using lineages as conditions instead of sex versus asex, and we're able to kind of compare all of those together, or separately, I guess. And this is what I'm showing you here is a set of 400 significantly differentially expressed transcripts between these lineages. And this is 400, just kind of a random sample. There were about 5,000 total that had significance between any two lineages. And what we see with these expression profiles is that population, lineage, and reproductive mode all seem to influence the expression profiles. So we have areas down here that are influenced by population. So up here, each of these are the lineages that I use. Red corresponds to asexual, blue is sexual, and there are lakes of origin I have up here as well. So these three lineages all originate from the same lake, Alexandrina, but this A1 asexual and A1 sexual are more closely related than to the A2 sexual. And we have selfie, sexual and asexual, and sexual or asexual Sarah. And we see that these three Alexandrina lineages kind of have shared expression. They have shared regions of expression profiles that show that they're all from the same population. And up here we have a selfie one as well. Lineage-specific signatures, like in selfie here, and in fact this region down here shows a lot of lineage effect on the expression profiles. They're quite different looking across. And then there's areas that seem to be influenced mostly by reproductive mode. Up here the two sexual Alexandrinas have lower expression for these transcripts, while the asexual has higher. And in fact, this whole region up here seems to show influence from reproductive mode. So the three sexuals have lower expression, while three asexuals have higher expression. And these are the types of transcripts that we want to try to identify and analyze further, because we think that they may be more important to reproductive mode and differentiating between sex and asex. Now, to some extent, looking at these heat maps, it leaves a lot to the imagination, and we have more questions sometimes after looking at these. They can be a little confusing sometimes. So what we wanted to do was try to identify some of the more important transcripts that might give us a clue into what is going on between the sexuals and the asexuals. So what I have here are each sexual lineage, and these are the transcripts that were always expressed higher in a sex versus asex comparison. So this here is 225 transcripts that were expressed higher in this A1 sexual lineage. They were expressed higher compared to every asexual lineage of snail. And ultimately we only see a few shared across that were consistently expressed higher in every pairwise comparison of sex to asex. So we've been working on running these sets of transcripts through BLAST2Go to find what they hit and what types of functions are related to them. So of these three that were expressed higher in every sex to asex comparison, we have these three BLAST hits, and they don't seem to have any clear reproductive significance. They're kind of general regulators of gene expression and transcription factors. So we weren't really sure what these actually mean. They're just they seem almost random to us. Another kind of interesting thing we found was that there were some transposable elements being expressed in these shared sets. So there was there was an RNA-directed polymerase from the mobile element jockey that was shared between the two Alexandrina lineages, as well as the selfie and Alexandrina 2. Now again, we don't know if this is actually part of the maintenance of sex, but it is interesting that we've found transposons being expressed in only sexuals, not asexuals. And the go terms for these that are associated with these with that transposon, again, kind of general replication functions. Now, if we look at these same sets from these same types of sets from the asexuals, so that is each of these cases, they were expressed higher in the asexuals. So here, these 247 were always expressed higher in the Alexandrina asexual lineage. And again, we looked at those 6 that were shared every time, that were expressed higher every time, and 2 of them didn't return any BLAST hits. When working with non-model organisms, you tend to find a lot of transcripts that don't BLAST to anything. And again, we found genes that didn't really strike us as these types of reproductively significant proteins that we were kind of thinking we might find. Again, they're just kind of, we did find some mitotic function in these, and but again, just some general kind of regular types of genes that you might find in any kind of set of genes that are being expressed. So to summarize, we see that lineage, lake of origin, and reproductive mode all do influence gene expression profiles. The asexuals tended to express more transcripts at higher levels than the sexuals, but ultimately very few of these were consistently expressed higher in asexuals compared to sexuals, or lower. So there are very few as well that were expressed higher in the sexuals. They were there though. We were finding some that had this sort of these consistent expression that were consistent from our expression test. So we think that this may suggest that there were unique mechanisms for each of these transitions, and that the sort of subsequent evolution after these transitions are different every time. But these loci that we've found are ones that we are going to try to characterize further with the molecular evolution from our genomic sequencing of these lineages as well. And with that, I'd like to thank the rest of Maureen Neiman's lab. I'd like to thank the University of Iowa and the iBio graduate program that I'm in, and everyone else who has helped along the way with this project, and the Sequencing Center for their help. So thank you. Any questions?